The lab that I run is primarily doing research in molecular imaging. Molecular imaging is sort of defined as creating images from a human being, but those images are based on imaging biochemistry and changes that occur with disease. So for example, you can think of an MRI, but what the MRI is really showing you is an image based on differences in proton density. What we add to that is to get information about the biochemistry that's occurring. The goal of the laboratory is to non-invasively image the biochemistry that predisposes the development of a tumor, for example. So a change in gene expression, change in biochemistry, and then the growth of a tumor. When you see it with a conventional MRI, the tumor is fairly large. When you see it with molecular imaging, it's detected much, much earlier. One of the major impacts that we're using molecular imaging to achieve is how does one remove a tumor surgically? Every surgeon that I've talked to will tell me that always there's a point within the surgery where they're making a guess, and that's at the edge of the tumor. Am I cutting normal tissue away, or am I still cutting diseased tissue away? So one of the aspects that we're doing is using the changes in biochemistry of these tumors to identify the difference between the normal and the tumor margins. We have currently been developing a lot of techniques where we can apply optical imaging agents and with a special camera view the tumor, which is now glowing in the surgical field, and then remove all the glowing cells, leaving the normal non-glowing cells. Breast cancer, lumpectomies are removed from women as the standard of care, and about 40% of those women have to come back because the pathology tells them that some of the cancer was left in the patient. Our goal is to impact patients in a number of different cancers by making the surgeries of a higher quality and more precise. Genomics and proteomics can identify pathways that the tumor is exploiting to grow. They do a needle biopsy, take out a few hundred to thousands of cells, and then analyze the genomics on those cells and understand the pathways. But that's a small portion of a very large tumor. It has differences in and among areas of that tumor that exploit different pathways. So now you could imagine visualizing an entire tumor using molecular imaging and having a red region, a green region, and a yellow region representative of different pathways that the tumor is using and different drugs that should be utilized to treat that tumor.